Okay, round two. Let's get the notes going. Okay, so the second part of this video lecture, we're going to talk about seedless vascular plants. Whereas earlier we talked about seedless non-vascular plants. Okay? So, just a couple of quick things that unite all of these seedless vascular plants. Well, number one, they have vascularity. So that's obvious. Two, they're dominated by the sporophyte this time. Sporophyte is the main plant in the life cycle of these plants. And here we actually get into heterospory. And I'm going to asterisk that because I have a little tangent I want to talk about in terms of the heterospory. Okay, so let's start with the vascularity. In these vascular plants, they have tubes that can transfer water from the roots upwards to the leaves. And in these plants, in vascular plants, they have two kinds of main cells that make up these tubes. You have the vessel elements, which are less common in these seedless vascular plants and more common in the seeded plants versus the tracheids, which are present in all vascular plants, both seeded and non-seeded. But they dominate the vasculature in seedless plants. And the actual nature of these tracheids, you have these cells that look like that, and they grow up and they eventually die off. What's left is their cell wall, which is thick and made out of lignin. Again, remember this not only allows them to make these tubes that transfer water, but also it gives the plant structure, right? The lignin in the cell wall allows the plants to grow taller because they're so thick and sturdy. So you have holes all throughout the cell walls of these plants, allows water to flow through this kind of amorphous grouping of these tracheids. The stacking of these different cell types are important, and we'll see later on the vessel elements, they stack up nicely like this because they have these walls that allow water to go right through them in a column-wise manner, so they're more efficient. For now, we're going to focus on the tracheids. These are the guys that, again, dominate in the seedless plants. And what this vascular also kind of brings forth is leaf biology, right? Leaves, as far as you need to know, are these fleshy pads of photosynthetic tissue. So the main point of the leaf is to gather light energy for the plant to make sugar. And supplying this leaf with water is vasculature. And there's two kinds of leaves you really need to know. Well, there's probably only two kinds of leaves in this designation that we're talking about, but you don't really need to know the details of like how you describe them. We'll describe them based only on their vasculature. We have the microphylls, which has one leaf vein going inwards, versus the megaphylls, which have a bunch. And these are the main leaves that we see in most plants. Microphylls mostly dominate the leaf structure of the club mosses, quillworts, and the horsetails. Okay? Let's move on. I don't really need to go back into detail about the sporophyte being the dominant plant in vasculature, in plants with vasculature. Just know that all vascular plants, so everything later than mosses and liverworts, they all are dominated by the 2N sporophyte. I think we just went over that already, but just to drive that home. Okay. Now let's go into heterospory. So looking back, I'm not exactly sure the way this is defined. I know rigidly it refers to having a larger spore and a smaller spore, which turns into a male gametophyte and a female gametophyte, which then gives rise to eggs and sperm. But some mosses, like we went over last week, or maybe actually just in the previous video, they have spores that kind of look the same, but they give rise to female and male gametophytes that give rise to eggs and sperm on separate plants. And I looked this up online, and there's no real end-all be-all answer to this. So I'm going to go ahead and say that because the spores are not different sizes, that the mosses are still homospores, or maybe just not any designated spores at all. But for sure, this is the rigid definition of heterospory, where you have different sized spores giving rise to different gametophytes, which give rise to different gametes on separate plants. Okay? So we'll stick with that. And this rigid definition of heterospory is found in some of these seedless plants with vasculature. It's present in all the different groups that we're going to look at to some extent, but still the main way of life is homospory, where you have spores that are the same, give rise to bisexual gametophytes that have both sperm and egg on the same plant. This is the dominant life history in all these seedless vascular plants, even though there are some that are heterospores. You'll see as you get more and more advanced in the seeded plants, they're all heterospores. Cool. So let's get into the actual individual organisms that we're going to look at. Let's start with club mosses and quillworts. So there's basically two body types you need to look at, the club mosses and the quillworts. Let's talk about the club mosses first. These guys are dominated by leaf structures that are consisted of microphyls. And we'll see the club mosses, they kind of look like this. So from the top, you have these microphyls that kind of whirl around the main vein. And from the side, they look like this. And the ones that you need to know are all branched at the nodes into twos. Right? Every node has a splitting pattern of two. The quillwort, it's pretty distinct. Nothing else in this lab looks like it. Just take a picture of it. I think it was Iocetes. That was the genus. I'm pretty sure. So remember, it was the one that was underwater. It had these long leaves, these long microphyls that just extended outwards like that. And it'll be in a tank. So if they decide to put it on the lab practical, you'll know that whatever looks like this is in water. It's going to be quillwort. So in this group of club mosses and quillworts put together. But the club moss is very distinct based on the branching pattern, and they're small microphyls that just cover the leaf or cover the stem. Their life history, we looked at the tips of, let me draw that out again. All right, we'll draw out the sporophyte like that. And we had the microscope slides that looked at the tips of these guys, remember, and under the microscope, they looked like this. And what these little dots are, sporangia. So this is where all the spores are made and held, and they're at the base of these leaves, right? And these leaves are sporophylls. Phylls being, I think it means leaf, so spore leaves. This structure is kind of a loose, I guess, predecessor of cones. As you get more and more and more later into the plant groups that we'll look at, these leaves get smaller and tighter, and eventually they just form cones. And everything that is cone-like is made out of leaves with spores at their bases, are all called cone and cone relatives. Are all called, what are they called? I'm blanking. They are called strobili, or strobilus. So this terminology will apply for the gymnosperms, as well as the club mosses and the horsetails, which we'll see in just a little bit. Okay, in these club mosses and the quillworts, and this is an emerging theme throughout all of these seedless non-vascular plants, their gametophytes are really small. Small, real small. A lot of the gametophytes in this group are actually underground and non-photosynthetic. So their entire point is just to poop them out into the ground when they germinate from the spores, and they just make gametes. Gametes find each other, make a zygote, and make a new 2N sporophyte, which is the dominant plant. So just in terms of identifying features, I think the club mosses and the quillworts are pretty distinct, not too hard to identify. In fact, I think most of the plants in this lab are pretty distinct. It'll get a little rougher in the seeded plants, but for now, the identification I think is going to be fine. Now we'll move on to horsetails. The defining feature of identifying horsetails, they have these really distinct segments. And these segments can either contain microphylls that are pretty big, or these microphylls can be reduced to a point where they're simply just these scratches on the segments themselves. Okay, at the tips of these plants, when they're making spores, they also have strobili, but they are more, the leaves are more tightly packed together, they become scales, essentially, and they just look like pine cones. They're a specialized unit used to protect and make the spores, which is the purpose of cone. Again, reduced gametophytic stage. We didn't really get into too much detail about that. Just remember microphylls. And the segmentation really, really distinct. You'll be easily able to identify these guys from everything else in this lab. one last thing to note, they're both for, I guess, structural reinforcement and to deter predators. The hollow tube that makes up the body of this plant actually has glass in it. So I remember Alice told us to pinch the main body of the plant itself, and you can hear some cracking. It's just like some of these grasses, for example. You actually have these little pieces of glass that reinforce the structure and prevent predation of these plants. Glass. Next, we'll get into the whisk ferns. Again, really, really distinct. Where they have megaphylls, or they once had megaphylls, and you can see that in these like little bumps, these nodes of the main stem of the plant. But these megaphylls got reduced to a point where they're just gone. And the sporangia are at the base of these shrunken megaphylls. So just see these big balls that are on the main stem, and that's really the identifying feature of the whisk ferns that you're responsible for in the lab. And their gametophytes are all, well, at least from what we've been told, are all underground. There's a note made of this in the slide describing the whisk ferns, so I'm going to assume they're mostly underground and very reduced. And they are non-photosynthetic. All right, so that's all good. The ferns, they are pretty, I would say for this lab specifically, it's easier to identify what's not a fern rather than what is a fern. But just some basic features of their biology. They have the fiddleheads. They're immature fronds. They grow out, unfurl their fronds. They grow into these mature megaphylls. At their base, all these megaphylls are connected by a lateral stem called a rhizome. This rhizome is present in other plants that we talked about earlier, but it's especially notable. You can actually see this not just underneath the soil, but above the soil. It can be this big fuzzy mass in the ferns. Okay, and at least in the ferns, roots come off of the rhizome, and those root system, right? This is a vascular plant, takes up water, brings that water to the leaves. Okay. What is really, I think, important for you guys to know in terms of fern biology is to identify the sporangia and the different versions of the sporangia in these ferns. So when these ferns are ready to produce spores, if you'll recall in lab, at the base of the leaf, at the bottom of the leaf rather, you'll have these dots. And each one of these dots is a collection of sporophytes. Sorry, sporangia. So these units that are making spores. So you'll have a sporangia, which are a collection of spores, and these will be clustered together to make these. Why can't I scroll? Okay. Make these little mounds full of sporangia at the base of the leaf. And this is called a sori, or a sorus. And something really important to note in the structure of these sori are whether or not they have coverings of very specialized tissue to protect them. These are called indusia. And there's a pretty wide variation of different indusial types that you looked at in lab. You have like kind of this dried covering like this, or you can have these lips that are in the middle of the leaf that kind of just bracket the sori. But they're, as long as you have any kind of tissue that seems to be surrounding or on top of the source versus a naked source, it just looks like this very homogeneous dot, you'll know that that is an indusium. With the exception of one plant that we looked at, I can't remember which, I think it was, I don't remember the genus, but there's a fern that looks like this. Really, veination is really thin. You have these little nice heart-shaped-ish leaves coming off of them. I think is an adiantum, I think. But there's only one that has a false indusium. And just go through your photos, remember this plant, and you'll know it's got a false indusium. But how you know it's a false indusium is that it's not a specialized tissue that covers the sorus like this or like this, but it's just the margin of the leaf. So you have the leaf like this, and the leaf actually just wraps around. So on the bottom of the leaf margin, you'll see these brackets like this, and they're just covering the sori. So just the leaf margins wrapping around. Leaf margins wrapping over to protect the source. Great. So what do I want to say to wrap up ferns? Oh yeah. When the spores in the sori germinate, they make little gametophytes. And I think this is true, again, for all these non-vascular, in all these vascular seedless plants. I think all their gametophytes are referred to as prothali, but I'm not sure. Prothali, prothallus. But definitely the fern ones are. And you'll remember from the sea ferns, they have these little heart-shaped guys that have both antheridia at the bottom and archegonia at the top, but also they have male-only gametophytes, and assumedly female-only gametophytes as well. They just differ in structure. It just depends on the fern, depends on the environment it's in, right? Prothals. Cool. To wrap everything up, what you really want to take away from this previous lab, one is to just know all the features that allowed these plants to jump to land, how they became more terrestrial than their algae relatives. And again, those two charts at the beginning of the lab notebook, as well as in your lecture slides, will go a long way to understanding the biology of these adaptations. And then secondly, really just be able to identify the plants and then identify the different life cycles of the plants. If I were to give you a sporangium of a club moss, right, from the strobili, you want to know that that's a club moss strobili versus a horsetail strobili versus the sori of a fern. All these different structures you need to know. But they're all pretty much related to how these plants reproduce and grow and divide and reproduce again. So know